That's much better. Good morning. Our beautiful girl from last week has shed her skin and she's looking really lovely. See how colourful she is now? Have a look at this. There's the skin, all off, all gone. Pretty awesome. We'll just wait a couple more minutes for some people to come through and then we'll get going today's experiment. So I'm going to leave Calypso out for a walk this morning. She's a bit cool, but she's shed her skin, which was why she was dark last week, and now she's very bright again, really light, lots of beautiful colours. So if you have a look on the back here, there's a perfect number 3 on Calypso. Awesome, eh? So she's a carpet python. They call her a coastal carpet python. But she's going to go for a nice walk this morning while we're out doing a couple of experiments in the yard. I've got a couple of cool ones for everyone today, and I reckon everyone at home should be able to try these themselves. Yeah, pretty excited. Alright, so off goes Calypso. She's going for a wander. We are going to play with a couple of ideas today. We're going to go through a couple of theories, hypotheses, laws, facts, explain what those words mean a little bit, because the way scientists use those words is entirely different to the way we use them in general talk. So we'll go through those words a little bit today, and we are going to run an experiment with a fish tank and some Coke bottles. Hopefully that connection picks up. And then we're also going to run an experiment with a bunch of flowers today. So I'll show you what we're going to do, and then we're going to go through in a bit more depth what's behind it. Hey, Joey. Yeah, mate, I'm sure she does. She's been an absolute gem too lately. She's really lovely. All right, so today, 2 experiments. One is animals, one is plants. So what we're doing is last week we did observations, so we were observing things in the yard, and we come up with a couple of ideas around what might different things do, like bees. Bees might go to different coloured flowers and things, so we're going to test that hypothesis today. And the other thing we're going to do is we are going to test germination of beans in different controls. So we've got, what we'll have is beans planted in normal soil, fertilised soil, and soil with coffee in it. Because there's a bit of an idea that coffee might help the gardens, but from the studies I've seen, coffee looks like it actually inhibits growth of plants. So I'm going to take you over. Let's have a look at our flowers today. So I've picked some flowers already. We've got a lot of different colours, so you'll be able to find different colours around your yard. And I'm going to go and show you the bees again real quick for anyone that missed the bees last week. And so we've got beautiful little honeybees. What we're going to use today is this ladder here. And we're actually going to put flowers up here and see what the bees do. So our bees are just there in that hole. They're flying around. We're going to go over them soon. So actually what we might do is we might set the bees up first so we can get them going, and we'll check on them from time to time and just do some recording, and then we'll come back to our experiment that everyone can run at home. So our next experiment here is I have a fish tank. If you don't have a fish tank, little bottles like these will be just fine. So we're going to separate our treatments for our beans. So we'll have fertilised normal soil and soil with coffee in it, and I'm actually going to run one with dog poo too, see what dog poo does to germination of seedlings and how they grow over time. So we're going to use this fish tank for one. I've already got our soil here, and it looks like our mate Calypso's coming back this way. But today, let's actually run the bees and flowers first. So I'll go through with everyone just quickly. I'm just going to pop that on the stand now. I'll go through with everyone what the idea behind this is. Now, last week we talked about animals being able to see different colours. So humans, we can see blue, blue, green and red. I think that's right. Bees can't see red. So they can see whites and blues and ultraviolet, whereas we can't see ultraviolet. So bees and humans, we both have 3 photoreceptors, but they pick up different wavelengths of light. So today we're going to test the idea that bees can't see red and they prefer white, yellow or ultraviolet flowers. Now, if we have a look at some of our flowers here, now you'll see on the inside of some of these that there is a yellow bit in the middle and there's white on the outside. Now that's actually called an ultraviolet nectar guide. So for bees and other insects that can see ultraviolet, that little bit of yellow in there might have ultraviolet spectrums on it, wavelengths that bounce back, and bees can pick that up. So they're going to be attracted to that because they can see that quite easily. Now I'll show you an example of what the difference in how bees see a flower as to how we might see a flower. So this is something I printed out a little bit earlier, but you can see there on this side, you can see how humans and other animals might see a flower, pretty normal, and this might be how bees see a flower. So you can see how that colour there, this is the same exact photo just split, you can see how that colour there has a little bit different in the middle, and for the bee it might perceive it entirely different, so very bright, very obvious. Now bees also have a really, really fast colour perception. And so their eyes are phenomenally fast. They actually have 5 eyes. So bees have 2 complex eyes right here, and then on the top of their head they have 3 what are called simple eyes or ocelli. And so they have really fast eyes, and they can actually pick up the difference in between individual flowers. So whereas we wouldn't really see too much difference in between individual flowers, they can see the difference in between them. So they can see one's brighter, one's more colourful, lots of different things. So really, really cool. We are going to take these flowers up to our little stand, and what we're going to do is run a little observation experiment. So when we run an experiment, what we're doing is we are essentially testing an idea that we've come up with. So in science, the other day we were observing different things around the garden here. We noticed that bees potentially like white flowers more. So today we've had our observations and we've come up with some ideas in our head. Now with these ideas, you can go back to your computer and research them, which is what I've done with this picture here. So for anyone that's just joined us, we're talking about how bees see flowers differently, that they might prefer a certain colour of flower over another colour. So we're going to test that out here. So we've got our observations. We've done a little bit of research on the computer or something and seen what's out there and from other people. And so we think that bees don't like red because they can't see it, and they prefer whites, blues and violets and ultraviolet. So now we're going to develop a hypothesis. So this works for anything. So my hypothesis with the bean seeds we'll try later is that coffee isn't good for growing, but fertilizer is. So that's my hypothesis there. I've observed different things around, and now I'm going to test that hypothesis. So in science and ecology, there's a thing called scientific method. First step is always observation, and you write your notes down in your little notebook. From there, you get some ideas, and you turn those into hypotheses. So very different to a theory. So when we're talking about the word theory in common culture, everyone says a theory is just an idea, people are guessing, but a theory in science is actually something that is very robust. So a theory is a hypothesis or a bunch of hypotheses that have been tested over time, and they actually are very robust. It's pretty substantial in terms of science. So theory, we're looking at things like Darwin's theory of natural evolution, natural selection, that kind of thing. So that's more of a theory, whereas when we talk about it in common culture, theory is just a word that's used very wishy-washy. It's lots of guesswork involved, so the two entirely different words, so that's really important. So today we've got our observation, which is, bees made like different coloured flowers. Now we've got a hypothesis, is that bees like whites or violets or blues. and don't like reds, and now we're going to test that hypothesis with an experiment. So there's your level. You've got your observation, you've got your hypothesis, and today we're going to do some testing by experiment. So there's a few things that you've got to, I'm going to sit on this log here, that you've really got to take into consideration with experiments. One is randomization. And you've got to remove variables, because there's so many different factors that can come into play with, I'm just going to grab these flowers, with these different flowers. Now, ideally we want to move every variable except colour, but in the perfect world that's not real. So you can see these two flowers here, very different in structure, anatomy, so we have to make some assumptions that the structure may not change our experiment today, and that's always the way it is with some of these. Ideally, if you could get several flowers with exactly the same structure but different colours, that'd be perfect. So assumptions are some of the things you have to make. So we assume that the flower structure isn't going to come into play, that our presence isn't going to affect the bees today, and a few other things down there. Now I can see a flower down there, and one of the flowers happens to be a violet one, violet white, which is a colour that flies like for flowers. So you always have to make your assumptions. Lots of variability, so you always got to take into consideration what else might be impacting the experiment. So the temperature of the day, whether the flowers on one end are in a cold temperature and the others are warm, or if one's in the sun and one's in the shade. So you have to make things as even and perfect as possible before you really start to test your experiment. So remove your variables, make a couple of assumptions that things might be the same, and then lots of repetition. So repetition in science is key. You have to have a robustness about it. Now, the bare minimum for most studies is actually 10. So the rule of 10s, you have 10 samples for each category that you're testing. So let's say we have 10 flowers of the white, 10 flowers of the blue, and maybe we actually repeat that experiment 10 times. That's when you get that level of scientific certainty that's good for science anyway. So we mightn't have all that repetition today, but repetition's really important. So if you can repeat something a couple of times, it takes out that variability a little bit more. So let's go up and check out our stand. We'll set up our flowers. Now this is going to be a little bit tricky for me, but I'm going to take my notepad over and some flowers, a couple other things, and we'll see how we go. We might take a couple of trips to get it over there. So I'll set you up so you can watch the bees or something in the meantime, and we'll chuck some flowers up. So it should be pretty exciting, I reckon. I'm keen to see how this goes. And then we're going to get our second experiment going. Now, everyone can do this at home. I reckon you'll just about be guaranteed to have flowers in your yards and guaranteed to have bees in your yard. So you can set up a little test plate like this as well, and we'll see how we go. So I'm just going to raise this up a bit higher. We've got a couple of things going on here, so we'll see how we go. I'm sorry it's upright today. Actually, I tried to make it landscape the video, but something happened with it was like making it really like it was a GoPro nearly. So I'm just going to go back and grab these flowers. I'll be back in a couple of seconds. If crossbreeding of flowers have changed the way bees pollinate, those type of flowers are more likely to self-pollinate. I'm not an expert in it. I'd be guessing. It looks like I'm going to have to set up a umbrella here to control some of the volume, because we got roadworks out the front and you might be picking up quite a lot of that sound, so I'll try and block it. I was hoping 10 o'clock would be a good time to do that, because that's when we've been taking lunch each day, but yeah, sometimes it changes. So I'm going to set up this umbrella here to hopefully block out a little bit of that noise anyway and catch mine. I'm just going to grab these flowers. I'll be back in a sec. I'll think about that question actually too. All right, beauty. So what I've got today is a board. You can see I've already got some sections drawn across on it, and I've got 5 of those, so I'm going to put different flowers in different sections. We'll try and keep colours pretty close together in the middles, and then we'll actually count the bees that go to each one. Now, the way I'll do this later is I'm actually going to set up a time-lapse camera on this, and afterwards I'll go through on the computer and count how many bees in each photo per every 5-minute interval, say. The problem with if we do it right now as a continuous study, what you might see that might affect your data is that a bee first goes to blue, but then he goes to red and then to yellow. So it's hard to kind of say what are we going to do with that, and you need a lot of detailed statistical analysis later to break that down. So an ideal way to do it would be every 5 minutes going back and seeing what bees are on what flowers. And that's what I'll do with a camera. I'll set the camera up to take a photo every 5 minutes, and then on the computer later I'll actually count how many of which species and yada yada. So I hope that this umbrella here is doing its trick with the volume. We'll see anyway. Oh, she's going to break. We'll see how we go, folks. It'll be interesting. Nice. Cable's familiar. Yeah, this is in Cairns. I'm still not sure about the cross-pollination. I imagine it would have changed the way bees pollinate, but they're still getting nectar regardless. They'll go for whatever nectar's out there, I reckon. So, yes and no. To move their bees into a give box. Now, that's something you probably have to do a little bit of reading online about how to get them into a give box if you're talking about where you can harvest honey and that. Definitely something that you'd want to look up on some of the bee websites online. But from what I understand, you often need a queen before you can take it to somewhere else. So you need a queen or one of the reproductive casts to be able to start that. I'm not sure how you'd entice them away. So let's set up this board and we'll see what happens there and get some nice flowers in it. I'll take you down with me. Alright, so we've got different colours of flowers here. Let's get our whites in there. So I've got a couple of whites that I picked up this morning while I was walking Ruby the dog. Let's put them down there, and I've got some pawpaw flowers as well here. They're very white, so we'll chuck them on the same one. Beauty. Alright, so we've got some whites. Now let's go, oh, I've got some more whites actually. So I got these as well, and I think they look pretty cool. So let's try them. Chuck a few of those in and see what happens. They're quite pretty. So I've got some more whites here. Wonderful. So the whites are on the board. All right, let's go purples. We've got some purpley violet coloured ones here as well. They're pretty cool. Now remember, anyone can do this at home. Really simple, really fun. What you might have to do is you could either watch it and take notes as you go, or just every 5 minutes come back and count how many bees, or take a photo actually, because the bees might move around. Take a photo and then count how many bees are there every 5 or 10 minutes or 60 minutes. Whatever you do, just make sure it's consistent. All right, we've got some beautiful yellow ones. Chuck the yellows on. Now obviously you'll have to try and place the flowers upright so that their pollen is showing. That'll be really important for it. All right, so our board is growing. All right, we got some reds too. We got some good reds, solid. Chuck them on. And I've got some more purples. Actually, more pink, sorry. Let's try a pink. We'll use a pink one. And I'll chuck a couple of these purple ones in because they're a little bit different to the others too. Now, what I tried to do is have an even amount in terms of volume of flowers, just so there wasn't too much bias. And that's something you've got to control as well. So here we go. We've got our board. We've got our different colours. Now, all I'm going to do is I'm just going to chuck this up here, and I'll show you where the nest is. And we'll come back in about 5 minutes, and we'll see how many bees are on each flower and have a look. But I'll show you where we are. So here's our flowers, and just there is our bee nest. So these are little native bees. They're stingless native bees. They're called sugar bags. I nearly got the name the other day, but they're called, their genus is Tetragonula. So it's just changed recently, but they're a wonderful, cute little bee, and they have no stings. There's about 11 species of native stingless bees in Australia, and so they're quite sweet, really lovely. And today we're going to see, hopefully, if they pick any of our flowers. So that's what we're looking at, just there. So their little nest is just up here. and we'll come back in about 10 minutes and we'll check that out, and we'll run our other experiments while we're doing that. I'll take these other flowers out of the way so we don't impact that. Oh, and the other thing is I want to show you a picture graph of what the bee colour might look like compared to ours, what their spectrum covers. I'll chuck this back on our stand just here, and then I'll show you. You ready? Alrighty. Hey, good morning, Teresa. Good morning to Neil and Tyson. Hey, Brett Wright. Hey, folks, there's a bloke there called Brett Wright. He's a helicopter pilot flying over in Bhutan at the moment. He flew the King of Bhutan around yesterday. How amazing is that? So he's stuck over there for a while until he can make his way back, the poor bugger. Good on you, Brett. Glad you can join us, mate. All right, so what we're looking at here, what I was talking about before is bees can see different colours to us, so they can see ultraviolet, which is why they can see this ultraviolet pollen guide here. Those colours are different to these colours. Same flower, just split in half. That's how we might see it, that's how bees might see it. Now this here, humans can see most of these colours here, so red all the way across to violet and blues. But bees can't see the reds here, but they go from, you know, nearly yellow, orange, all the way over to UV over here, so ultraviolet. So different areas, the different eyes can pick up different things, but the different eyes have different photoreceptors in them. So the bees have the photoreceptor for ultraviolet, whereas we don't. We both have 3 photoreceptors to pick up light. Ours are a little bit different. We can pick up red. So pretty cool. And you can find these online. I'll put some links up later for them too. So for now, I should check where Calypso is actually. I don't think the neighbour would appreciate if the snake went into her yard. Oh, here she is. Okay, let's have a look. There she is, just down in there going for a walk. So some things out. I better remind me to check on her soon, someone please. Can you send me a message in about 10 minutes saying check on Calypso to see where she is? Otherwise she might sneak off. All right. So today, the other part of what I'm going to do is later on, this is called a field camera or trail camera. You can set this up to either sense motion, so sense animals coming by, senses movement, and it'll take a picture. We use these in field studies a lot. Or you can set them up to run a time-lapse function. Now, I've got heaps of time-lapse videos online of lagoons over 2 years in Cape York filling up and going down, cows and horses that have died, decaying, and a cow takes about 7 days to decay. From a full cow to just bones up in Cape York. It happens really quick. So time-lapse cameras are phenomenal. You can see how things go. I actually got a fungus growing in the yard the other day that grew and then died and went back down, and you can make a cool quick video of it. But I'll use this later to check on the bees and monitor them and do that count. Now, all you want to do when you're taking your notes and counting your bees is really just something simple like that. Put your colours up the top there, red, and just put tallies in it. 1, 2, 3, 4, really simple. And then maybe do it, actually what we could do is you could trial 3 times a day as well. So you could do maybe an hour every 5 minutes in the morning, or half an hour every 5 minutes in the morning and count the bees. And then you could do half an hour every 5 minutes at lunchtime, and then half an hour every 5 minutes at the end of the day in the afternoon when it's cool. Now, from what I understand, I think bees are more active when it's cooler as opposed to in the middle of the day, for pretty obvious reasons. But yeah, we'll see how we go. All right, experiment number 2 is plants, and we are going to use different things today. So when we're talking scientific method, you often have a control. Now, a control means you have something that has had no treatment on it, so there's no effect. So our control today, what we're going to be doing is I've just got some climbing stringless beans that we're going to use. These little guys here. Now we're going to plant them in just normal soil. Plain soil, nothing done to it. So that is what we call a control, because there's no treatment done. Now the other 2, or potentially 3, the other 2 in the fish tank just behind me there, they're going to have, one's going to be the control, so no treatment. Second is going to be with some soil from the worm box that I showed you the other day. It has the worms in it. They're breaking down all of our scraps, and there's lots of fertilizer essentially in there. So people use worm tea or worm juice as a fertilizer that you can spray on, which is this stuff here. So that's a bottle from that is definitely not orange juice. Don't drink that, you'll choke. But this is fertilizer, worm tea from that box the other day. I've probably got about 10 litres of it, and I'm just not putting it on the garden fast enough. So really good stuff. So that's going to be our second treatment. It's going to have soil from the compost in there, which should be rich and really good nutrients. And then the third is going to be our little friend here, coffee beans. So these are coffee grounds that have gone through, the coffee's been drunk, and these get thrown out. Now, what a lot of people do is they'll chuck them in their compost or in their garden. And I've often wondered what the effect of coffee on your garden is. Now, I don't know what the reason is, so that's our hypothesis. We're going to check if coffee beans are good or bad for plants. We're going to do that by germinating bean seeds, which germinate quite fast, and monitor them over time and see what their growth is. Now, I'm going to plant as many bean seeds as I can fit in that fish tank behind me, and I'm going to have a second set in the bottles there, which everyone can do if they don't have a spare fish tank, of course. Much easier, and you get to separate the treatments a bit better there too. So we'll use coffee. I've seen some studies online, I've done a little bit of research that suggests that coffee beans are actually bad for your garden. They might make it acidic or something, but whatever it is, they often stunt or impede the growth of the plants. So I'm going to run you through what we're doing today, and I'm just going to start putting some of this stuff in. We'll see how we go. I might take us down another level here. We'll get right down on the ground, get our hands dirty and have some fun, and try our second experiment. Actually, let's go and take a sneak peek of the flowers and see if anything's changed there before we get into it. We're not too far away, so let's have a look. All right, here we go. Ooh. I don't see anyone yet. We'll come back soon and we'll check again later. So it might be one of those things you have to go back quite often, or just sit at and watch for the first little while. But I reckon in the morning when it's cooler, probably would be one of the best times to do it. All right, so here is this one. Where are we? This one here. This is just standard soil. That one down there is our compost soil. And so all we're going to do is we're going to mix this up. I've already moistened it to make it nice for the plants, and then we're just going to add it into the fish tank there. So I'm going to come over here and we'll set up, and we'll start doing our experiment. Beauty. Now, send us some questions, and I can... Oh, check Calypso, thank you. Here she is. She's done good. Thanks, Shane. Shane Ross Photography there, watching on. All right, so here's Calypso. She's going for a walk. Off she goes. Don't tell the neighbour. All right. She's going to come and hang out with us in a minute. I'll probably get a fright. She'll lick me on the foot or something. Awesome. All right, yep, send us some questions through, folks, while we're getting set up here. Now, I am sorry I mentioned it before, but we had to go longitude or upright today. The camera, whenever I turned it over to do landscape, was actually playing up quite a bit. It was being really distorted. Oh yeah, interesting. There you go. So yeah, there's another experiment there, folks, from Melinda that you could trial. Melinda says that she's heard that coffee has benefits for disease control, and the same with tea leaves. So whether that's a matter of the coffee being in the soil or making a spray and spraying it on the leaves, don't know. So it's one of those things you can try. A lot of people also try and use chili spray. So making I guess an oil or a spray from chili and spraying that on their leaves as an organic parasite protector, or whatever you want to call it. But so there's different options there we can all test. Today, let's fill up our little guy here. Alright, I'm going to try and make this a bit quicker by chucking those in there. Alright. Now with that soil there, if you look at it, it's quite fine. I actually put it through a sieve just to remove all the big bits, so there was none of that, no more external factors kind of playing with the germination, like big rocks or any funny bits playing with it. So we'll fill these up. Now I better fill, what I'm going to do here is, excuse me, I'm just going to grab a bucket behind me so I can mix things in, because I will mix the coffee beans in down the end one there as well. Actually, I can probably mix them in there. Alright. Now I've got this is the compost one. I'm going to save a little bit of that compost one for our bottle as well. Alright, there we go. And then might actually, while I'm doing this, I'll just sprinkle a bit of this in. So this is our coffee beans. So our last one there, we're going to chuck coffee beans in. And I'll mix that in a little bit more as we go. Now let's get a bit closer. What we're going to do is we're going to use a pencil. We're going to put as many beans in here as we can, and then hopefully in what does it say, about 5 days they should germinate. Germination 7 to 10 days. I think beans germinate a little bit quicker up this way. So Teresa has a question. She's wondering if you place coffee grinds in your compost and then added compost conditioner, would that help with the pH levels? I've heard in the compost it's a similar effect, so I'm not sure if you added compost conditioner. Yeah, potentially. Yeah, and that only answers though if it is the pH that's affecting the growth of the seedlings and not something else. So potentially, yeah, absolutely, good question. All right. So I'm just going to use a pencil. Always read the packet because the packet knows best. So it says 25 mil, so 2.5 centimeters or an inch. Let's see how many. We might try and get 5 or 6 in each of these little segments here. So let's see how we go and see how coordinated I am. Actually, what I wanted to do, and the reason I was using a fish tank, is I want to put them really close to here so I can actually watch the roots germinate as well and take a video of that with a time-lapse camera. So I'm going to adjust those a little bit. There we go. How's that looking? Yeah, you can see them. Perfect. Wonderful. So there's the first 5. Now what I'll end up doing with this whole batch actually is I'll plant more rows further back in here as well. And that'll give me a little bit more scientific rigor and repetition. And so I won't just have the first 5 that I can see with the video camera. I might have about, let's say 20 all up, which is a pretty good sample for each treatment. So 20 is more than 10. 10 is that cutoff. You don't want to go less than 10. So I'll have 20 in there for each treatment, which is really good. And that also accounts for if a few of your seeds weren't viable anyway and they were never going to germinate, that you're not just down to 1 or 2 to compare, because that's not quite scientific, because there's also individual variation within species, and so that could account for growth. So by repetition, you actually try and accommodate for that. We'll keep going here, and then we'll go over to the bottles as well and try the bottles. Let's see if this pencil works a bit better. Beauty. Awesome. Now the other thing I'm going to do too, folks, so you don't forget, is write on here what each one is. So this is compost, this is coffee, and this is standard. Or actually we should name that one control. Alright, our last 5. Beauty, so that's done. Awesome. So you can see the beans in there. We can see them from the side. So what I'll do later is, again, I'll once I'm done using this on the bees, I'll actually use this on the beans here. And over about probably, I'd say 20 days, I'll have that camera going just to watch the growth. Later on after the video, I'll go through and I'll plant more seedlings down in there. So I'll have about 20 each. And then I've also got here just bottles, folks. So I've cut up a bunch of bottles. I've got, what have I got, 4? So I've got 4 bottles, and I'm going to do the exact same thing. I'll do one quickly now as an example, and then we'll probably wrap up and go to questions. And questions can be on anything. So if you think of anything that you didn't get to ask in the turtle session or the observation session, shoot your questions through, because we'll have some questions and a bit of free time. Today's a nice short easy one. So I've already drilled a few holes in these so they can drain if I water them. If you have a fish tank and you do the fish tank, just be mindful that the water can't drain, so you don't want to oversaturate the soil in there. Now there you go, that's the home version. If you don't have a fish tank, perfect, just cut a couple of clear bottles. Then all you want to do, same thing as a fish tank, chuck them around the outside so you can see them grow. Chuck a couple on the inside too. Now just remember how many you put in there. So I've already put in 5, and later on when I put more in, I'll count just to make sure I know how many actually germinated, and I can compare that level too to see if the coffee affects germination as well as just growth. So we're going to chuck our seeds in here. Now beans are really good because they've got quite a large seed, a lot of fat and protein and growth stuff in there for them, so lots of energy to grow. So that's why they're usually used in these kind of experiments, these quick play around ones. Alright, so you can see in there I've got a few seeds planted in there. Now that's my first one, so that was just the standard, that's called my control. That's my standard experiment, and then I'll do the others with the rest. And then I'm actually, if you missed on earlier, I'm actually going to use this last one. I took my dog for a walk this morning real early, and she laid a mighty old nugget on the road. And I thought, I wonder what the nugget, the dog poo, would do to plants. Because if you're at home and you've got to take the poo out of your yard and put it in the garden, maybe it works as a fertilizer, maybe it doesn't, I don't know. So later on when I take the dog for a walk again, I'm going to collect that poo, and I'm probably going to put some gloves on, and I'll mix it up with some soil and put it in here, and I will see how that affects germination and growth rate as well. So that'll be a cool little one, and anyone can try that if you game, and maybe you got a pair of gloves. Maybe borrow mum's dish gloves. Don't tell her, and then don't give them back. All right, so that's our experiments today. So our first one was bees. Where's our paper? So for anyone that came in late, we'll go back and check on the bees again before we go. Anyone that came in late, we're doing 2 experiments today. One is animal and one is plants. The way we work, just to rehash, and ecology. I should have explained ecology. So ecology is the study of interactions between animals, plants, and organisms in an environment and in relation to their environment as well. So it's the study of everything interacting and how it interacts and why it does what it does. So that's the basis of ecology. What we're doing now is a scientific method or a scientific study, an experiment essentially, to test some things we saw from observation that we turned into hypotheses that we're going to either try and prove or disprove today. So our idea with bees is that they can see different colours. They can't see red, but they can see ultraviolet, which we can't see. Now, again, looking at this picture, flower, that's what it looks like for us. That's what it might look like for bees. So they can see this ultraviolet pollen guide in the middle. So I think today we might see more bees on the white and purple flowers than the reds and the yellows, but we're going to confirm that by counting. So that was our first experiment. Our second experiment is just here. So you can see our fish tank there. On this side we have soil mixed with coffee. Then we have our compost soil, which is essentially fertilizer. And then we have our standard, which has no treatment at all. It's just normal soil. So what I did, that was just soil from the garden that had been mixed in with a bit of potting mix ages and ages ago. Yesterday I got a sieve and I just sieved it. Now I'll show you what I mean by sieved. You can use your old, if your dad or anyone in your family's got one of those gold kind of sieves. So it's just one of these. It's quite coarse. If you go any finer like this one, it's too fine, I find. The slightly coarser one, so that's about a 5mm mesh, and that works really nicely to get that fine soil. So we'll see how that goes. When we're done here, I'll go through the rest of those pots and fill them up too, and I'll fill you in on the Facebook page, on the Cockatoos Facebook page, every couple of days when things germinate. All right, let's sneak up to our bees now. All right, so it doesn't look like there's anyone there yet, but that's okay. We'll keep coming back and we'll keep checking, and hopefully it'll work. So we'll just keep monitoring that over time. Here's our bees just here. Now I put them close because apparently bees can really only see very well within about 60 centimeters. And without that, they actually use a few other things to navigate. One is they can see polarized light, so they can see patterns in the light in the earth that they use to go from A to B, and that's how they find their way home. They do that little wiggle dance too to tell the other bees where they go. And they can smell as well, so they can use smell to find their flowers. So let's go over here, just out of curiosity. Now there's going to be a bit of roadworks in the background, but I'm just going to see if there's many bees. All right. There's a couple of flies mating up there, but actually there's not many native bees up there as there was this morning. So there you go. I just made an observation that our bees are probably more active when it's cool, and when it's warm they're staying in there. Now the other day, quick little one is, yes, Calypso's still free. I better go and check on her. We looked at our butterfly vine here, and we finally had one of the Cairns birdwing butterflies hatch into a caterpillar. So one of the eggs hatched into a caterpillar, and where are we? There it is. I'll try and bring it a bit closer. That's it there. It's gonna be a bit hard. I'll try and make that a bit easier. Let's see if that's any better. That's it there. So it just directed its little glands at the front. They're full of smells and lots of yucky stuff, but it's bright, so it's got yellow and red on it, and that's to deter predators. So what it actually does is it eats that vine over there that's got toxins in it. It eats that vine over there that's got the toxins in it, uses those toxins, takes them into its own body, puts them in those spines there with bright colours saying to birds and other things, don't eat me, you're not going to like me, I taste like crap, and it might be poisonous, so stay away. Awesome. All right, let's see if we've got any questions, folks. Send your questions through. Hey, g'day Aaron, AC, hey Summers. Check on Calypso. Go on. Ooh. Ooh. Where is she? She's on the trot, I betcha. Ooh. I'm gonna have to find her. Ooh. I'll find her. I better peek over the neighbour's fence. Nah, she must be in here somewhere still. I'll find her anyway. She's got me on my... Oh, here she is. Look at this. She's a darling. There she is. She's just going for a stroll. Awesome. Found the snake. Hey, Gail from Omeril. How are you, mate? All right, folks, if anyone's got questions, shoot them through. Oh, chuck a few magic beans. Yeah. Yeah, that'd take us to a better place than at the moment, I reckon. It'd be much more fun up there in the clouds. All right, folks, any questions, send them through. They can be about crocs, snakes, the turtles we did the other day, the plants and the bees we did today, observations. How'd people go in the gardens at home too, by the way? Did anyone find anything cool? I had a few cool stories come back. And did anyone find out why a frog doesn't have ribs, or what the idea behind a frog not having ribs is? Yes, Joey, I've lost her a few times, mate. I know she doesn't go far. She's real calm, so pretty confident she doesn't go outside the yard. But yeah, sometimes she goes for a little hide, and then I panic after about 15 minutes of not being able to find her. So, folks, the other day one of my questions was, I'll sit here for a bit. We've still got a few minutes anyway for anyone that wants to stay on, but it's pretty much question time now. We've done our experiments. The other day I asked the question about frogs. Now, frogs don't have ribs. If you look up frog skeleton on Google that, and you'll find that all they have is tiny little vertebral pieces left behind, but no ribs going around. Now, what I was asking everyone to find out was why frogs might not have ribs. So I wanted you to think about what frogs do. And as ecologists or people that study these things, that's something we often do, is we wonder why something occurs for a certain reason. We put lots of ideas out there, or hypotheses, and then we start testing them. Now, there's a few reasons why people think frogs don't have ribs. One of them is that they're ground resting, so they don't need the structure to support their body. But then things like lizards and snakes would be the same, right? So I'm not a fan of that one so much, but there's one reason why frogs don't have ribs that I really, really love. And think about what frogs do a lot of. Now, it might not be true, but I think it's a really good hypothesis, hard to test and hard to prove. But think about what frogs do a lot of. The way they move isn't by walking, they jump, right? And so lots of frogs are always jumping. Now imagine if a frog had 15 sets of tiny little ribs, because they can't be big because they'd be too heavy. Now, if they had 15 pairs of ribs on each side and they were jumping all the time, now if a frog jumped from the first story down to the ground and he had 15 pairs of ribs, there's a fair chance each time he did that, one of those would break. So potentially over time, they've evolved to lose their ribs. Potentially also so they can squish in little places, absolutely. But then you've got to think about other animals that have that ability too, like snakes and lizards and all those sorts of things still have ribs. So questions, there's always so many questions, but one of my favourites is, why do frogs not have ribs? Potentially because they jump a lot, and if they did, they would break them. Now, because they don't have ribs, it means they can't breathe the way we do, because our ribs form a structure, a solid structure, and then we have a diaphragm inside. Now, the diaphragm pulls, makes a negative vacuum, pulls, so a low pressure, and then the air goes into our lungs and then back out. Now, frogs don't have ribs or a diaphragm, so the way they breathe is by buccal contraction here. So you'll see at night time, if you have a look at frogs, their throat expands and it's pushing air into their lungs. The second way is through their skin, so they can actually gas exchange through their skin because it's kept moist. That's why we have to be careful when we touch them as well. And the third way is that at the back of their mouth they have a way of respirating, essentially gas exchange. So breathing doesn't always mean breathing the way we think about it, but breathing can be gas exchange. Now, a really cool thing, I did it yesterday, actually known it for a lifetime, but insects. So insects also breathe through their skin. They have pores, kind of like pores, where there's gas exchange through their skin. Now, if you put an insect underwater, it doesn't drown. Now, the reason for that is, and I haven't looked at it for a long time, so I could explain this very coarsely, is that you put an insect underwater and watch it in clear water, and you'll notice there's this tiny silvery sheen around it, and there's air trapped right around its body. So you'll never drown an insect, maybe after a couple of days or something, but short term, put a March fly underwater and all you'll see is this silver lining around it that's actually air trapped around it. Now, it does that by heaps and heaps of little hairs right around its body to trap that air in there. And so I can actually still keep breathing underwater, which is really cool. So breathing isn't always what we see as, it can be through the skin, it can be lots of different ways. So always something to be mindful of. Yes, yeah, that's right, Teresa. I'm pretty sure toads don't have ribs either. Exactly, yeah. Awesome. So any other questions? How's everyone? Hey everyone at home. I'm getting warm, as you can see. Hey Joe, JC, how's it going? G'day Trisha. Hey Jig-Jag. Hey Elizabeth and co. Allspin might have given us an answer for how to separate or to get the queen in. I just can't expand that at the moment for some reason, so I can't. Oh, here we go. Allspin Maslow said with that B question before about separating, that if you moved some of the comb into a long box, leave the box open for the hive for a few days and the queen will move into the box. There you go. There's an answer in the comments about that B question before. Brilliant. JC, you clown. Rightio, here we go. The jokes are coming, the questions are over. Do I still do the sea turtle work? No, unfortunately. I did that for 6 years. It was incredibly rewarding, but also incredibly hard work. It wasn't just the work you saw in the videos the other day. There was a lot of fencing and long hours, long days, lots and lots of work around that. So I don't do that anymore. I love it, and I still try and talk to the guys that are doing it, but essentially my job was to train other people up to do the work. So I didn't have to be up there. So the idea behind a lot of that is in Cape York, with the Aboriginal Ranger groups up there, they don't really want to see a lot of scientists, or not that they don't want to see them, but they would rather have their own guys working than scientists doing the work. So my job was essentially, as a scientist, was to train the guys up there to do my role and then only provide information and feedback on that afterwards. So it was really good. We trained the guys up. There's some amazing turtle dudes up there now from the Aboriginal Ranger groups, so that was really cool. Yeah. All right, any other questions? I hope today was a good session, folks. Actually, what I want to do is, if anyone has a topic for Thursday that they would like to suggest and would like me to talk about, please send one through. I don't have one for Thursday yet. I've got heaps and heaps of topics on the list there that I can go through, but I haven't done one because I wanted to put it to everyone else to see if there's a topic we could talk about in terms of ecology, animals, nature, research, that sort of thing. So if anyone has something that they would love Thursday's session to be about, let me know and we'll do up a yarn for that. But yeah, I've got a whole list of other things, lots of exciting stuff. I better go and check on Calypso. Thanks everyone for coming. Big love, stay safe out there. Take care of all your family and be loving and fun. Alrighty. Goannas. Oh, we could talk about goannas. Alright, I'll send you a message, Trisha, and let's see what we come up with.